Hello students, welcome back after the break. You are listening to your very favorite Dr. Sandeepan Sengupta, who is an assistant professor in microbiology. So we were discussing, before I went for the break, we were discussing for the first part of my lecture, we were discussing about methyl-directed mismatch repair. So as you all must be knowing now, so what is the process all about? The process, what does it do? It deals with the repair of rare mismatches left after the process of DNA replication. And what is actually the step which carries genetic information for the entire process? It is the first step of the process which carries genetic information for this entire process of mismatch repair. The process of mismatch repair can be divided into three different stages and consists of different events, three series of events. The first stage is all about the hemimethylation of the hybrid DNA, which particularly involves methylation of the parent or the template strand. In the second stage of the process, a series of proteins called MUT proteins, that is MUTL to name MUTL, MUTS and MUTH come into action. And in the third stage of the process, the mismatches are repaired, or the rare mismatches left out after the process of replication are repaired. So the first stage, that is methylation, actually directs the entire process and governs the process and carries genetic information according to which the process of repair is carried out, and the rare mismatches, or which carries information to direct the process of repair. So the first stage of the process is hemimethylation of the hybrid DNA, which consists of one strand of the parent or template and the other newly synthesized daughter strand. So the first step is hemimethylation, or the selective addition of methyl groups only to the parent or the template strand of this hybrid DNA molecule, but not to the daughter strand. And this, the way after the parent strand is methylated, this methylated parent strand directs the two more steps of the process and governs the way the mismatches are repaired in the template strand, in the daughter strand. So that is the first step of hemimethylation, which acts as the carrier of genetic information and which is the director for the entire process, is being carried out by an enzyme or is facilitated by a bacterial enzyme called DAM methylase. So what does the enzyme do? It specifically adds methyl bases to the N6 position of adenines of the 5' GAT sequences present in the ORC region of the template or the parent DNA. So immediately after replication, following this process of hemimethylation and activity of DAM methylase, the parent or the template strand is methylated at the N6 position of the adenines in 5' GATC sequences present in the ORC region, but the daughter strand is not, or is devoid of methyl groups. And during this lasts for a few seconds and minutes, and this hemimethylated parental DNA directs the process of methyl repair. So the second step of the process of methyl repair involves the work or the combination of an action of a series of proteins called MUT proteins. So in the second stage of the process of this mismatch repair, or the methyl-directed mismatch repair, it has been aptly titled because the methylation is the director of the entire process, and the way the G5' GATC sequences in the template strand or the parent strand are methylated acts as that is director for the remaining part of the process for the other two stages. That is, in the second stage after methylation, that is immediately a series of proteins called MUT proteins come into action. The second stage is all about the activity of MUT proteins. There are three very important proteins in the series called MUTL, MUTS and MUTH. Now, two proteins, that is a complex made up of MUTL and MUTS, binds to the methylated GATC sequences sequences encountered in the parent or the template DNA. Now next to this, what happens? The MUTH protein binds to MUTL part of the MUTL-MUTS complex. So the following, that is the follower, that is next to MUTL-MUTS complex, the follower is MUTH protein, which binds to MUTL part of the complex, that is bound to methylated template DNA or methylated parent DNA at the N6 position of adenines and the 5' GATC sequences present within the OSC region. So what happens is, what is the use of MUTH? It has an endonuclease or nucleolytic cleavage activity or an endonuclease activity which comes into action only when it encounters methyl groups bound to GATC sequences in the OSC region of the template DNA. So once MUTH binds to MUTL and encounters that is methyl groups bound to the parent or the template DNA, it introduces its activity, that is endonucleolytic activity or the activity to cleave called DNA, called endonuclease activity, gets activated and comes into play and introduces a cleavage, but not in the parent but in the daughter strand. So MUTH, the endonucleolytic activity or that is endonuclease activity of MUTH cleaves the daughter strand of the hybrid DNA, that is according to the methyl groups present in the GATC sequences, that is of the parent or the template strand. Now what happens next to this? After a cleavage is introduced in the daughter strand, the mismatches are repaired in the final stage of the process called mismatch repair according to their location respective to this cleave site or the cleavage or cut that has been introduced in the that is daughter strand. So that is a different set of enzymes come into action to repair the mismatches which are present on the 3' site of the cleavage that has been introduced in the daughter strand, and a completely different set of enzymes and proteins come into play in action to repair the mismatches that are present on the 5' site of the cleaved daughter strand. If the mismatches, now let us see the differences, or let us peep into the different proteins and the molecular mechanism or underlying mechanism which comes into action and repairs the mismatches on both the sides of the cleaved site. If the mismatches are present on the 5' side of the cleavage site in the daughter strand, then they are repaired as a result of the activity of a complex made up of several different enzymes and proteins. Now at first comes into action exonuclease proteins called exonuclease 1 or exonuclease X. This exonuclease 1 or exonuclease X comes into action only for the comes into action for the mismatches which are present on the 5' side of the cleaved daughter DNA. And what if the exonuclease 1 and exonuclease X altogether will degrade the mismatches or the DNA portion are part of the daughter DNA. After a part of daughter DNA has been degraded by exonuclease 1 and exonuclease X, new DNA is being synthesized in that portion to fill up the gap by DNA pol 3, and the remaining nick is sealed by DNA ligase. And with this ends the process of the repair of mismatches which are present on the 5' site of the cleaved daughter strand. Now what about the process occurring at the 3' site of the process occurring for the mismatches left present on the 3' site of the cleaved daughter strand strand. The mismatches which are present on the 3' side of the cleaved daughter strand are repaired as that is when a series of different proteins called Rec-J nuclease and exonuclease 7 come into action. So especially for the mismatches left on the 3' side of the cleaved daughter strand, that is the portion of mismatches containing the daughter strand, of the part of the daughter strand containing the mismatches is cleaved due to the activity of Rec-J nuclease or exonuclease 7. And after this, again the enzyme, that is in order to fill up this, that is the gap that is generated as a result of cleavage, DNA Pol 3 comes into action. DNA Pol 3 synthesizes new DNA and the gap is sealed by DNA ligase. So therein lies the difference between the repair of mismatches that is occurring on the 5' side of the cleaved daughter strand and that occurring on the 3' side of the cleaved daughter strand. The difference lies only in the way the mismatches lying on the 5' side of the daughter strand and the mismatches lying on the 3' side of the cleaved side of the daughter strand are removed by the action of exonucleases. So if the mismatches are on the 5' side of the cleaved daughter strand, they are removed due to the activity of exonuclease 1 or exonuclease X. And if the mismatches are present on the 3' side of the cleaved site of the daughter strand and are on the 3' side of the cleave, that is site of the daughter strand, they are removed due to the activity of exonuclease 7 or Rec-J nuclease. Then next to this cleavages, the other steps remain the same. That is the gap that is generated as a result of removal of the mismatches is being filled up by synthesis of new DNA that is done by DNA Pol 3 and ligase finally seals the gap or the nick, and with this the mismatches are repaired, and with this ends the final step of the process of methyl directed mismatch repair. So the process occurs in 3 different stages. The first is methylation, which specifically methylates the parent or the template strand but not the daughter strand. The second stage involves the activity of 3 proteins called a series of MUTL proteins which come into action and introduce a cleavage MUTH which has an endonuclease activity. It gets activated on encountering the methyl 5' GADC sequences in the template strand but introduces a cleavage in the daughter strand to repair the mismatch bases. Now in the final stage of the process of repair, depending upon whether the mismatches are present on the 5' side or on the 3' side of the cleavage introduced in the daughter strand, the different proteins come into action and they repair the mismatches. If the mismatches are on the 5' side of the cleavage introduced in the daughter strand, they are being degraded. That part of DNA is being degraded by exonuclease 1 and exonuclease X, and if they are on the 3' side, the part of DNA containing the mismatches, or that is the portion of the daughter DNA, is being degraded by exonuclease 7 or Rec-J nuclease. Then following this, new DNA synthesis occurs as a result of the activity of DNA Pol 3 and the nick is sealed by DNA ligase. Now that is all about the 3 different stages. The process completes in 3 different stages, and with the repair of the mismatches on both 5' and 3' side of the cleaved site of the daughter DNA strand ends the process of methyl directed mismatch repair. Before I went for a break, I left a very important, that is a very interesting question for you all. I asked you all to work out or research, that is to find out the homologs of MUT series of proteins which are found in eukaryotes are analogous to the MUT proteins found in bacteria. In bacteria, and particularly the system that is well known for E. coli and related bacteria, the MUT proteins are MUTL, MUTS and MUTH. So the homologs of MUTL and MUTS but not MUTH have been found to particularly exist in humans and yeasts and other eukaryotes. So what are these homologs called? The homologs of MUTS and MUTL are found to exist, but MUTH have not been found in eukaryotes, and these homologs in eukaryotes correspond are similar to structure and function to those of their proteins present in prokaryotes. That is, most of these are the homologs of MUTS protein of prokaryotes, and accordingly has been named as MSH2, MSH6, MSH, that is MUTL, S synonymous, H is homolog to MSH6. That is, these are the proteins which are found in eukaryotes like yeast, and humans are analogous to MUTS protein found in bacteria or MUTL protein found in bacteria, and are involved in the repair of mismatches in case of eukaryotes. So that is the end of the topic of methyl-directed mismatch repair. At the end of today's discussion, I have a very interesting activity for you all. Now, will you please take a look over, that is, while going through the methyl-directed, you can consult the book Learning Principles of Biochemistry for getting into more detail of the process of methyl-directed mismatch repair, and please specifically go through the role of these homologous proteins which are found in eukaryotes that are homologous to that are analogous, in fact, that are similar in structure and function to the proteins called MUTS protein found in prokaryotes. That is, please go through the role and the function of MSH2, MSH6 proteins which are found in yeast and eukaryotes, and please have a look over the mechanism and the way they carry out they work to remove or to repair the mismatch bases in eukaryotic DNA. So stay tuned for another lecture. I'll be coming up with another process of DNA repair system. See you all. That is for the next. See you all on another day, on a very fresh day with a new topic. That is, till then, take care of your health. Stay tuned and keep studying. Goodbye, students.